talk about what is aging as a biological process, what types of data can be used and analyzed for this, also about the RNA sequence data and particularly the data set that was used for this research. We'll also talk about how to analyze such types of data because it has its own characteristics. And then we'll try to build linear and non-linear models for predicting age and try to select some of the features and interpretate the meaning of those. So first of all, personally, I for a long time thought that aging is an inevitable process, and I thought how can I deal with it? But while I was thinking, many other people were actually doing something. So they did lots of research on what is the biological cause of aging, can aging be delayed or even stopped? They thought about is healthy aging possible, because we are not happy not just because aging exists, but because it causes many related problems. And aging in general is not, it doesn't feel healthy. Then also we think about what types of biomarkers can indicate age. So of course we can look at the person and understand whether this person is aged or not, but we know that different people age differently, and there are real biomarkers that can say something about it, and those biomarkers can also be possible targets for some gene engineering that will be possible, I hope, in the nearest future. Also, one topic is can nutrition slow down aging, and actually there are many research projects around that. And of course, there are many aging-associated diseases, and we think that probably once we understand what aging, we might be able to somehow evade those. And actually the area of science which researches all of those questions is gerontology, but the part of it which is responsible for biological part is called biogerontology. And this research was conducted in the field of biogerontology. So currently we know several things about aging. We know that there are at least 3 levels on which aging occurs. It's a genome level, cell level, and organism level. So on the genome level, what happens is that DNA repair mechanisms break, and that's why there are cells that are damaged, and some tissues are also damaged. We also know that telomeres that are on the ends of our chromosomes starting to be shorter, which is also one of the, I would say, side effect of aging. Then we also know that our genome consists of a coding part and non-coding part. The coding part codes proteins that have some functions, but the non-coding part, the epigenome, it regulates the genetic expression. And when it regulates the expression good, there are no problems, but once this mechanism breaks, there are problems, and these problems actually also come with aging, with age. We also have on the cell level protein folding, mitochondrial function, stem cells, and on the higher level, we have the problem of cellular communication that happens in the incorrect way. And all of these processes, they cause or they are effects of aging. And I think that one of the main levels here is the first one, is the genome level. And in this study, we are going to talk about the genome level of aging. In particular, the genome part means that we should somehow analyze the DNA or RNA data. We can receive gene expression data from RNA sequence here, just a small part of what's going on, how to get the gene expression data. So you first have some kind of your biomaterial, then there is a nucleic acid isolation happening, then we have some specific gene-specific PCR, then this is actually the process of next generation sequencing that is very often used right now. And in the end, we have the RNA. And one of the outcomes of this analysis is gene expression data. And here we can see the little heat map, because gene expression is actually very often shown on the heat map, where we know that, for example, the red part means that the expression is high. And in this research, we had the GTX dataset, the open dataset created by Broad Institute of MIT and Harvard, which means genotype tissue expression. And the reason why this dataset was created, because our organism has different tissues, and it's interesting to know how the gene expression changes between tissues. And here on the right side, we have a picture of all the different tissues that were analyzed, from which we were able, the people who created the data set were able to retrieve gene expressions. And we can see that it's not only contains the tissue, but also subtissue. For example, for the brain tissue, there are more than 10 different subtissues. And the data set consists of two parts. The first one I would call a labels part, which is here. So we have our subject ID or donor ID, the person from whose organism the sequence was done. And we also have tissue. And here we can see that we have one person, but the sequencing was performed from different tissues. We also have the sex of the person. And what is important for us is that we have age bracket. Unfortunately, we only have the bracket or range, which means that we don't have exact number of how old the person was when the sequencing was conducted. It's because of the privacy, but still it shows us something, and it's enough for us to build a machine learning model around it. And what's important here is that we also have the ID of the sample. So we know the exact ID of this tissue and subject combination. So this is like the labels part here. The second part, very important, is actual gene expression data. The column here on the left is the one that we use to join this part of the data with the labels data set, which means that for each row here, we know the age from the labels data set from the previous slide. And each column here represents actual gene, and these are ensemble IDs of the genes. And the numbers here are gene expression data, which is in a transcript per million. So as you remember from this image, we have different nucleotide sequences that were mapped to specific genes. And here we just count how many were mapped for the particular gene. And there are different metrics how this gene expression number can be calculated. The one that is used here is called TPM, or transcript per million, so that it's actually a relative number. We calculate all of the transcripts for this sequencing sample, and then we just normalize it. What's also important is that in particular this metric, we also have normalization by gene lengths. And it's important because different genes have different lengths in number of nucleotides, for example. And it should be noted while calculating gene expression. The size of the data set is quite big in terms of the genomic data, because it's there is a privacy issue in the field, and this data set is one of the biggest open data set. So we have 17,000 samples and 18,000 features. So we have 18,000 genes to analyze. And the task for us is to find from those 18,000 genes which of the genes form this H bracket. So it's like more a formal idea of the task. So we have to predict the H looking at the gene expression. It's also interesting that for H we have 6 H ranges from 20 to 80, and we don't have exact ages, that it seems that this task looks like a classification problem, but in fact, as it was in many literature examples, the age is more continuous variable, because the class 50, 59 should be logically included into the class 60, 69. So it more really looks like a regression problem, and it was written about it in the literatures. That's why we decided to try to tackle this problem as a regression, and if something bad happens, then we can always try to do classification. Yeah, so the first thing to do is to look at the distribution of the data. As we have 18,000 features, I just took 20 random features, gene expressions, and built the histograms around. And we also don't know whether we should use linear or non-linear models, and the data looked like the linear model would not work good here, at least without any transformations. And there are 3 reasons for that. First, that we have unequal variances for each of the gene, so it's actually natural property of this data, because some genes can be more expressed than others, even not in relate to the age, but it's just that because genes are different. So we can see here how different the variances can be. We also see that all of the histograms are skewed to the left, which also differs from the histograms we should be getting from the data which is distributed normally. And we also can see that we have extreme values. So gene expression data cannot be negative. So all of the genes, the minimal value is 0. And we also can see that the variances are not just different, they are really different in terms of even one gene. So there are many extreme values here. And all of these reasons, it seems that we should think about the data pre-processing. But it's also important to know whether maybe we just selected those 20 genes randomly and all the other genes are good in our data set. That's why the Shapiro-Wilk test for normality was conducted for all 18,000 genes. And for no transformation, there were 0 genes that were thought as normally distributed features. That's why we had to do some transformation, and we performed the logarithm Box-Cox transformation with the 2 criteria for selection, for optimizing the parameter, and also one interesting, at least for me, transformation which is called Blom, or inverse normal transformation. And for the 3 random genes again, we can build the histograms and see how different they are for each of the transformations. And we can see that even from the pictures here, that the Blom transformations histogram looks really like a normal distribution. And there is a reason for that, because inside of it, first uses the ranks transformation, which makes all of the gene expressions distributed uniformly, and then using some special function, it converts uniform distribution to normal distribution. So even though we see it on the pictures, we again performed the Shapiro-Wilk test for transformed data, And we can see that the numbers did not really change for the 3 types of transformations. However, for the inverse normal, we have almost 1/3 of the genes which are now considered normally distributed. And it seems that this is a first assumption that we can make, that this transformation may lead to better results for the linear models, but we definitely need to check it when building a model. And so then, after transformation, when we know that our data is at least a bit closer to normal distribution, we have to choose a linear model to try. And we still have some more assumption of the gene expression data. The first one is that we know that some of the genes might be correlated because they are in the same biological processes. So the second is that we know that for sure not all of the genes would be biomarkers of aging, and we have 18,000 genes. So we need to combine 2 of those, and actually there is a model which perfectly does it. It's called ElasticNet, which combines both penalties, rich penalty, which would allow to have equal weights for correlated features, and it would also have lasso penalty, which would allow us to make 0 weights for those features that really not contribute to the model in enough way. So we use this model for one more reason, because there is a successful example when one more aging clock, but built on the methylation DNA, was built using the ElasticNet model. And here on the picture, we can see how good the model is. So we see age and some feature built on the methylation, and this aging clock actually still has the best accuracy among all the other methylation clocks. That's why it's like one more reason for choosing this model. So the schema for linear models would look like this. We do some kind of transformation. Then we know that our values, expression values, are more distributed normally. Then we have 2 ways. First, we can just try to build ElasticNet model using the transform data, but also we know that for the model might not still work correctly with correlated data, so that we can use PCA to ensure that our features are not correlated between each other. So we kind of want to help the model. And we can also use different number of components, because essentially we don't know which number of components would be the best. And then while training and evaluation, we first of all use the cross-validation to select alpha and L1 parameters, which are named for this ElasticNet model. And we also need to, it's just nice to know that the scikit-learn version of ElasticNet has the parameter called positive, which makes all the weights of the features that ElasticNet assigns to the features positive. And I don't know why, but it's true by default, so we should set it to false, because in our case there totally might be genes that would lower the estimates of age, and we need to know that. These are the results. So here we have multiple tests. We have, all these tests are for ElasticNet, but we have different transformation type and we have different features. First, we can see that the results from no transformation are the worst. It's something that we expected. We also have results for different transformation functions. We have results for all genes, for hundreds, thousands, and 10,000 principal components. And as a result, which is kind of really logical, is that the best accuracy on the test set that we get is from the inverse, normal inverse transformation, or BLOM, and 10,000 components. which is kind of something that we expected. But still, when we look at this number, 0.46, it seems to be not really a good accuracy, which definitely motivates us to try some nonlinear model and to see how they would perform on this task. So for nonlinear model, we still have some schemas to do, because there are many properties of the data that we know about, and we can use this in the nonlinear models too. First, we need to choose the algorithm, and here the LightGBM was chosen, I think, for at least 4 reasons. The first reason is that we all know how good this algorithm is. The second reason is that we know that our data set is quite big, and if we want to perform multiple rounds and trainings, we need to have some optimization, and LightGBM has the dataset function inside, which optimizes the data for the calculation it makes. One more reason is that our task is not just to predict aging, but actually to get biomarkers, get the features that are most important for the model. And LightGBM has this feature coming in the box, so we don't have to perform anything special. We just can call one function, and we can sort our feature by gain or split. And one more reason is that we still know about the correlation, and we know that tree algorithms are quite good at working with such features. So that's why this algorithm was chosen. But again, we want to help our model somehow, because we know that the size of the features is actually higher than the size of the samples. And even for such a powerful model as LightGBM, it's still hard, it might be hard to find some good patterns in such with such number of samples and features. So what we do is essentially training just a simple LightGBM model, and then we can get features weights using gain and split, and one more criteria about which I'm going to talk a bit later. Then we just not do this weighting once, we do k rounds. So on each round we just shuffle the data set and we get new weights, and then we only leave those genes that have non-zero weights on all the rounds of iteration. And here it's important about the number of rounds that you can perform. And this, first of all, this is a trade-off between how many features you want to select and how sure you want to be in the features that you select. So first of all, it kind of provides the stability of the features that you choose, but also the more rounds you perform, the less features you will have at the end, and it's not always that you want really small number of features. So what we do is that we perform this feature selection like many times, and at the end we have some number of genes, in our case let's say 200, and what we do is that we train a new model which have not seen the data before, but we train it only on this 200 of the features, and that's what is done here with this arrow. So using this new model and those selected ages, we predict ages, and at the same time we have a trained model to explain. Again, we do this because we know that not all 18 hundreds or thousands of genes are needed, and we want to help the model to find really those genes that for all iterations would have good weights. And such a schema leads to the following results. So we have here multiple tests. We have just a simple GBM model which has only one round, and it's trained on all genes, and we can see coefficient of determination, mean squared error, absolute error, and Huber loss, which just assigns a bit different weights to the anomaly incorrect predictions. So it's more like robust to them. We also have this 3 models of GBM, which were built using the previous schema. And from here, we can see that actually the coefficient determination is not really changing much, but all of these 3 models are much better than just simple GBM model on all of the genes. But we still see that the selection using gain has better mean squared error and absolute error. We also can see our linear model here and what was its accuracy. And we also have just a baseline model based on mean of the ages and a random model which would assign the age interval randomly, and we can see their performance, which is really bad. Anyway, now we have our model which is better than others, and we finally can get biomarkers from it and try to explain what those biomarkers mean. It's also nice to note, because of having so many rounds, you have to think about how you evaluate the model, because you shuffle the data set many times and you have to have some kind of cross-validation scheme for that. So here we had the GTX dataset, which was, and this is something that was performed during each round of the training of the GBM model. So we have 5 folds, for example, and each fold would be split into the training data, which would be used for training, and the test data. But test data should be split into 2 more groups. Because GBM model is I would say high-level model, the same as the neural network, it has a really cool parameter for where you should not explicitly state the numbers of the iterations GBM should do. So you do this it just measures the validation accuracy, and once the validation accuracy does not improve for, let's say, 5 rounds, you just stop the model. And for exactly this, you need to have your validation data set that would allow GBM to calculate that. But you also should have a clean test for which GBM did not calculate anything, so that you can be sure that this particular result is something that the model have not seen and have not used for any kind of optimizations. One more thing to look at as overfitting and how the models are learning. It's also cool that these models are generally from different nature. So here on the left side, we can see ElasticNet and how different its learning, its scores are for cross-validation and for training. And we can see that once we have more training examples, the cross-validation score grows, but the training score is falling down. It means that we are not really overfitting much, and we see that this gap between the results at the end, at the maximum capacity of our dataset, is really small, meaning that the model is not really overfitting. But what is also interesting here is that imagine that we would have more samples in the dataset, and in our case, the GTX dataset has, I would say, each 2 years, they have more, just 5,000 samples more, which means that we can easily imagine that our dataset is bigger, and sometimes in the future we would get this learning curve perfectly, maybe meeting it at some point. But what is important is that our cross-validation score probably would never go higher than 0.55. And it's not good for us, because we want our model to be adapted to the data. We want that, we want our model so that the more data we have, the better results we have. And probably from ElasticNet, we could not really ask for more than this score. But looking at the GBM, the picture here is really different. First of all, it's important to note this real gap between the training and cross-validation score, which means that probably the model is overfitting. However, here are the parameters that were used to control it, but still, maybe they should be optimized better to avoid this problem. But the positive thing here is that, again, if we imagine that we will have more data points, our model still could perform better. And even here, just because the split in the data was different, we can see that our coefficient of determination is higher than 0.60, which is better than we had in the table with results. We also can look at the accuracy dynamics. So here is our first model, the simple ElasticNet, which would not have any data transformation, and then we can see how much the result is better just using the transformation and PCA. Then when we add the GBM, we can see that again the result is better, and using the GBM and the gain selection criteria, and of course many rounds of GBM underneath, we can get the highest result for now. And the same for the mean squared error, we can see how it falls down. And actually this plot shows that it's not really that much important which model you're using. It's more important what you do with your data. So here we have absolutely the same model, just with one round, but doing the transformation and PCA, we can see that result just doubled in the accuracy. And maybe the same with GBM. It seems that it's really hard to make the GBM work better, but still you can do some feature engineering or selection, and you can achieve better score. Well, here we got our data, our features. So when we want to look at the biomarkers, we want to, of course, extract them from the best model. And in our case, it's a GBM and gain selection. So once we have it, as we are talking still about biogerontology, we not only need the list of the biomarkers, but we are also interested in how exactly they influence the age. And the biomarkers can be split into two classes, the anti-aging genes and the pro-aging genes, which means that anti-aging genes are some genes whose expression is falling while age is growing. And for pro-aging genes, it means that their gene expression is growing together with age growing. And we really want something to understand that. And there are many explainability libraries that could help with this. And here on the left side, there are at least 7 types of those. But some years ago, and you probably know about this library, the SHAP library was created, and in fact, what's cool about it is that it uses all of the methods that were existing before. So for example, and it also can explain not only one type of the model, but many types, like tree algorithms, some kernel functions, neural networks. And for example, for explaining neural networks output, they use underneath the deep leaf library, and for kernel functions, they use Lime. So it's kind of not, they have something new that they created, but they also, the power of the library is also in the fact that they use previously existing libraries for their library. And the assumption of all of these libraries and SHAP library is that actually they have the additive feature importance idea, so that when you have your model prediction here, it's F, you can explain this with your explanation model G. And how you're going to explain it? You're going to explain it as a sum of effects of each feature on this prediction. So for each prediction, for each row in your data set, let's say test data set, you could, for example, in our case, it's age. So our model predicted that some sequence is, its age is 25. And by using such a library, such a formula, you can split this 25 into, let's say, 200 parts. Yeah, if you, for example, chose 200 genes. And this formula is actually came from the cooperative game theory, which is a Shapley value. That's why the library probably is called SHAP, because if we look at the features and predictions and models from a bit other point of view, we can see that our features actually are players, and they are trying to cooperate and to get some, I don't know, let's say treasure. In our case, it's age. And in the end, for now, when we want to explain this, we want to know how many of the treasure each feature needs to take in a fair way. So that in our case, we want to understand what each part, each feature brings to our prediction. And there are several axioms that are in this game theory. Is that, first of all, treasure can be explained by some of the effects of each player. Then that when there is the same effect of the player, they should get the equal, I don't know, treasure. Then if there are multiple players there, and they have the sum of their effects, you can calculate it for each of those players, and that if the effect of the player is 0, it means that it should not get anything. So it brings us, and the theorem, the Shapley value, is that using all of these axioms, there is only one fair way how to calculate the splitting of this treasure, and this way is called Shapley value. So this is the theoretical part, which is underneath, part of it, these libraries and the SHAP library. And this is how this SHAP value is calculated. So here it's an effect of each feature, and we have a subset of all the other features, not including this ith feature. Here we have result prediction of the model when the feature I is included in the model, and here we have the prediction when the model is not included in the subset of feature, when the feature I is not included in the subset of features. And here you can calculate the difference between those predictions. And but you need also to account for the sets of the other features, like for other cooperations between features. That's why you should calculate it many times, but you can weight it by all possible subsets of the features. And at the end, you will get the number which in that particular cooperation this is the amount of effect that this feature should get. And here is just the same explanation, but not in the features and subsets, but like just for the more in theory games terms. And at the end, how this leads to our goal is that our prediction, our H, our F function result is the sum of the effects of each feature, where M is the number of features. And here is the result of, for example, we have the base value, which is 0, and we have this positive contribution, which are contributions of the features, which are marked with red, and we see that they push the prediction of the model towards the positive prediction. I mean, more the prediction should be higher, but there is still some feature with index 4 which have this pushing towards the lower prediction. And when we sum up all of these things, we will get our prediction from the model value. So, and here is how it could look like for a real case. This is taken from the paper, but it even seems that those are also genes. So we have some base value, and we also have our different effects of different features. We can see that our actual output from the model is 24. So our features in sum pushed it to the two more points here. And here we can see the numbers of the names of the features which were negatively had negative impact on the result, and that had positive. It's also important that this is the local prediction. It's also the plot, the SHAP plot, just for one row, one prediction. It's not the plot that you can build for, let's say, all your test data set. If you want to build something for the test data set, you can use another type of plot, which is called summary plot. So here we can see the genes. The color here is gene expression. So the blue color is low expression and red color is high expression. And here we have the SHAP value. What's really cool here is that SHAP value measures are the same as your target variable. So here we have -4 years, -2 years, and 6 years, which means that these first genes they are also sorted with the maximum impact on the model. And these genes, depending on your gene expression of this gene, your age estimate could actually be different on 12 years. And it's really massive range, I think. And we also can see that this gene, for example, is a pro-aging gene, because when it has lower value on the age, it also has low expression value. But when it has high expression value, it also has a high positive impact on age, so it forms age positively. We also can see here the result of also strong feature, but it's anti-aging gene, because when it has high expression values, it has a negative SHAP value, and when it has low expression values, it has the SHAP value is bigger. So using this technique, we can also actually look at the directions of each genes to understand how they impact our age estimate. We also have one more type of the plots that can be built in SHAP library, and that's the interaction plot. So here on the y-axis you have SHAP value, and on the x-axis you have your actual gene expression. And here, and the color can be just the color variable might be omitted. Then all of your points would be in one color. But if you want at the same time to look at the gene expression of some other gene, for example, here this one and here this one, you can also color that when you want, for example, to see how gene expressions of two genes correlate between themselves. We can see how differently genes could look like. For example, here, this is a anti-aging gene, and here pro-aging gene. We can see that difference in the plots. What's cool here is that we can not only look at this in the plots, as we have SHAP values that are real matrices, and the SHAP value matrix has the same size as your actual data set, but instead of the features, you would have SHAP values for features. You can calculate many statistics based on the SHAP values. And here, looking at this plot, it seems quite obvious that you can have some correlation coefficient, which would show you this dependency on the two variables, the SHAP value variable and actual variable. Well, you could say, if you want to calculate the correlation between age and feature, you just actually could calculate it. You can take the age column, and then you can take the the gene column, and just calculate the correlation. But the cool thing about the SHAP values and gene expression correlation is that when you have SHAP values, their value is isolated from effects of other genes. And if you wanted to calculate the correlation with age, the age forms with many variables, but SHAP values is definitely formed just by your feature, which means this is something that you can trust. And I used the Kendall coefficient because my data is definitely the dependency is definitely not linear. We can see it in here, and using this correlation coefficient, we can calculate calculate it and rank our biomarkers. So I had around let's say 500 biomarkers at the end, and I could rank them by this coefficient. We also have some genes that have really high positive correlations, but also some genes that have really high negative correlation. It's also and so there are like, these were two at the beginning of the list, and these two were at the end of the list, and there were many genes between them. But we also can always go to the literature, and when we find some biomarker, of course we may have some people with a biological experience who can tell you whether this biomarker really can have some impact or be impacted by age very hard or not. But we can also go to the literature and find whether someone already thought that this is a biomarker of aging. And for this 4 genes, I found links for all of them in the literature about biomarkers of aging. And it's really interesting, the function of these genes are interesting, because this one is for brain development, and this one is for DNA damage and tumor suppressions, and those two are positive correlated to age, which it's kind of hard to understand what brain development means, but there are many genes in one biological process so that this one particularly may have some negative influence on the development process of brain. But for these 2 genes, it's very logically, because this one is responsible for some things about cholesterol in cellular lipid removal pathway, and this gene has negative correlation with age, so that when age is higher, the expression of this gene is lower. Maybe it means that cholesterol is somehow not removed in a proper way, because gene expression is lower. And the same is for this gene, because it's responsible for protein transport. And again, it's important that all proteins are transported correctly. And when we have this negative correlation, it means that this transport, the quality of the transport maybe is lower with age, and I think that's quite logical that we know that this happens when we age. So talking about the current and next steps, it's just that this task, it's located here, is not somewhere in vacuum, so that these biomarkers that that we get from the GTX dataset, they actually are used to compare the biomarkers from the other dataset. Even more interesting task, where we try to find the pro and maximum lifespan genes, and where our dataset is not just human sequence data, but it's sequence data from from different species, and the target variables which we are trying to predict is not just the age of the animal, but actually the maximum lifespan that it can have. Because some rats may have just may live 2 years, human can live 100 years, but some whales can live 200 years. And in this study, we look at what genes could actually be somehow related to the maximum lifespan of the species. And then, for example, compare it with the genes that are anti-aging and see whether pro-maximum lifespan are also anti-aging, and genes that are pro-aging are anti-maximum lifespan. Also, as I said, for GTX dataset, we may have some new data in a year or multiple years, but we are not happy with the results. We want them to be better, and we can't wait for a year for that. So we can think about, and we already have generated few samples, so we can generate gene expression data using variational autoencoders and some other generative models, for example, generative adversarial network, to ensure the reality of the output the network produces. And also, it's important because with genomic data sets, there is a huge issue on privacy and on how hard it is to get data. And if we could really generate some realistic samples, it would not just help the GTX dataset, but many other genomic data sets. And just one more interesting idea that we have together with Google Summer of Code project is that, again, looking at this privacy issue, we use federated learning. We have a student who uses federated learning and DVC so that different hospitals may have their own data sets that they don't want to share. And for example, those could be RNA sequence data sets. And, but still, they understand that those data sets might help for some researchers. And the federated learning is a good way how you can learn on different small data sets which are located in different places. And in general, this research was conducted together with the System Biology of Aging group of Institute of Biochemistry of Romanian Academy, which is in Romania. And yeah, I think that's it. And if you have any questions, please ask.